Hey everyone, so this presentation is going to be on host lymphoid organs and tissues given through SCRUBS, which stands for Student Collaborative Resources for Understanding and Brody Success. My name is Peyton Shepard, and I'm a part of the class of 2028. Here is the SCRUBS mission statement. SCRUBS is a student-driven initiative that aims to develop supplemental resources for current and future cohorts that will pass through Brody. Members of Scrubs participate in a variety of subcommittees working to create resources for students by students. These resources aim to offer unique perspectives from students that have walked in the same shoes, developing resources that we wish we had been exposed to during our time in the course. The hope is this organization will become a staple of the Brody student body, exemplifying the unique collaborative community that Brody offers. If this is a mission that aligns with your goals and you have the desire to help those that will come behind you, as well as a goal to leave your mark on Brody as a whole, we invite you to join the team. Here is the Scrubs disclaimer. The resources that are included in this document are made by students and not the faculty. As such, there is a possibility for errors in our development, although this is mitigated via a team approach to development with multiple stages of vetting. If there is a contradiction with the coursework presented within your course, please go by the course documents. Please report any errors via the contact or error reporting link located on the Scrubs website. Additionally, Scrubs aims to supply supplemental resources. However, these are in no way replacements to the instruction of the Brody faculty. Use these resources as a supplement, but not as your primary source for course material. So the first thing I want to talk about is identifying our primary versus secondary lymphoid organs, and this is going to be important for later discussions that we're going to have within the course. So first thing we're going to look at is our primary lymphoid organs, and there's only two. It's going to be the bone marrow and our thymus. And then our secondary lymphoid organs are going to consist of your lymph nodes and your spleen, as well as your lymphatic vessels, MALT, which stands for mucosal associated lymphoid tissue, your tonsils, and Peyer's patches. So let's talk about the primary lymphoid organs' general roles. Again, our primary lymphoid organs are going to consist of the bone marrow and the thymus. And the number one general role is that these are going to provide hematopoietic inducing microenvironments for the differentiation of our lymphocytes. And more specifically, once the stem cells are differentiating into the T and B cells, this is going to occur in the bone marrow. Once they've differentiated from the stem cell into the respective T and B cells within the bone marrow, they're going to each go to different places to acquire their antigenic specificities. The B cells are going to remain in the bone marrow to go undergo this process, and then the T cells are going to go to the thymus to undergo positive and negative selection. And we'll explain this process at a later point in the course. So sticking with our primary lymphoid organs, again, it's going to be our bone marrow and our thymus. Once those stem cells had differentiated within the bone marrow to our respective T and B cells, that's when the cells start to separate and again to go acquire their antigenic specificities. And again, just to reiterate, the B cells are going to stay within the bone marrow to mature and undergo that process, while the T cells are going to mature within the thymus. And below is just some histological images to hopefully kind of connect the bigger picture to your other courses. This slide is also, again, just trying to connect some of the bigger picture ideas to your other courses. And as this presentation goes along, we'll go into a little bit more depth about the different parts of these structures and what is occurring in each. So now on to our secondary lymphoid organs general roles. Again, those secondary lymphoid organs are going to consist of our lymph nodes, spleen, lymphatic vessels, MALT, which again stands for mucosal associated lymphoid tissues, our tonsils, and our Peyer's patches. So one of the general roles that is going to be for our secondary lymphoid organs is that this is a place that is going to act as filters to trap antigens that come through the body. They are going to be sites of antigen processing and presentation. They are sites of interaction between antigen-presenting cells, T cells, and your B cells. Some of our antigen-presenting cells are going to include our dendritic or maybe our macrophages. And the next general role is the secondary lymphoid organs are going to retain antibody producing plasma cells, which is a subclass of our B cells. And the fifth general role would be that these secondary lymphoid organs are going to provide a reservoir of memory B cells and T cells for circulation. And those memory B cells are, again, just another subclass of our B cell category. So, for the secondary lymphoid organs, one of the most important ones you need to be able to understand is going to be the lymph nodes. So, the general structure of the lymph node is going to consist of a cortex, paracortex, and a medulla. So, first up is going to be the cortex. And as you can see here on this histological image, it's going to kind of be this outer portion is going to be your cortex. And then this middle portion here will be your medulla. So, for the cortex, it is going to be again the most superficial or outer layer, and this is going to consist of B cells primarily. The cortex is broken down into two separate parts, which is going to have a primary follicle and a secondary follicle. For the primary follicle, it's going to contain mostly follicular dendritic cells and small resting B cells. For the secondary follicles, it's going to have a central germinal center containing large proliferating B cells, your lymphoblasts, memory B lymphocytes, plasma cells, macrophages, and follicular dendritic cells as well. But the main concept you want to take away from the cortex is going to be that this is your B cell area. That's the big takeaway. Next is going to be the paracortex. And for the paracortex, that's going to be your T cell area. It's going to contain T lymphocytes and dendritic cells that have migrated from your tissues to the node. So again, your cortex is going to be your B cell area. Your paracortex is going to be mainly your T cell area, as well as some dendritic cells. Next up is going to be the medulla. It's the collection point for subcapsular sinuses prior to exit via efferent lymphatics, and this can contain a large number of antibody-secreting plasma cells. So the three main portions that you're going to have to keep track of are going to be the cortex, which is your B cell area, and that's going to be this outer portion here. And then your paracortex, which is going to house mainly your T cells, and then your medulla, which is going to house mostly antibody secreting plasma cells. And again, your medulla is going to be like right here in the center of your lymph node. All right, now that we've talked about the broad structure of the lymph node, we'll go into what the circulation of lymphocytes and antigens through the lymph nodes look like. So first up is going to be our lymph-borne antigens coming in from the surrounding tissue spaces, and they're going to pass into the subcapsular sinus via the afferent lymphatics. Once they do that, during their passage through the cortex, paracortex, and medulla, the antigen is going to end up being trapped by phagocytic cells, typically a macrophage, and it's either going to be degraded and destroyed there or broken up into fragments that might end up making their way out of the lymph node. For the lymphocytes and antigens that are exiting the lymph node, those are going to exit out the efferent lymphatic vessel, and they're going to re-enter circulation through your thoracic duct. So some of the broad points of this, again, are going to be your lymph-borne antigens are going to travel into the lymph node via your subcapsular sinus and the afferent lymphatics. They're going to travel through and potentially be stopped by and trapped by your macrophages. And anything that makes it past this point is going to exit the lymph node via your efferent lymphatic vessels to enter the circulation again through your thoracic duct. So transitioning from circulation of our lymph-borne lymphocytes, antigens through the lymph nodes, now we're going to discuss the circulation of blood-borne lymphocytes going through the lymph nodes. So for these types of lymphocytes that are blood-borne, they're going to enter the node through arterial blood flow. They're then going to enter specifically the paracortex of the lymph node via passage of postcapillary venules. These are also going to be termed high endothelial venules, or HEV. After they end up passing through the paracortex, the cortex, and then the medulla, these lymphocytes are going to exit via the efferent lymphatic vessel. And off to the side here is just a little bit of an easier flow diagram that might make it easier to understand. So again, the highlighted points are going to be the lymphocytes that are blood-borne are going to enter the lymph node via arterial blood flow. And these lymphocytes are then going to enter the paracortex of the lymph node through your high endothelial venules. After that, they're going to travel through the paracortex, cortex, and medulla to exit the lymph node via your efferent lymphatic vessel. So the next really important secondary lymphoid organ that we want to have a good understanding of is going to be the spleen. So some of our basic functions of the spleen are going to be that it collects antigens from the blood specifically. So it will not collect antigens that's within the lymphatic system. It's also going to be the site of the antibody synthesis to blood-borne antigens. So the big key here, takeaway, is that it's going to be collecting antigens from the blood and creating antibodies for those blood-borne antigens. Now, going through some of the circulation through the spleen, for those, again, we're just talking about blood-borne antigens. They're going to enter the spleen via the splenic artery, which is then going to empty into the marginal zone. Once they enter the marginal zone, it's going to be trapped by antigen-presenting cells. Typically, again, it's going to be your macrophages or cells like that, which carry the antigen to what's called the periarteriolar lymphocyte sheath, or PALS. And then here is a histological image that we'll go through on the next slide, kind of talking about the different parts of the spleen. Here's a more of a basic cartoon version of it. So sticking to the spleen, it's going to be broken down into three main parts. Two of the main areas are going to be your red and white pulp, and these are going to be separated by your marginal zone. So if we look at a zoomed in picture, we're going to have our red pulp all out here, along with our marginal zone that's right along this border here, just a little bit darker, and then our white pulp here. Now, what's going to happen in our red pulp is that it's a network of sinusoids populated with macrophages that remove your sensient red blood cells. So this is basically the garbage disposal for your red blood cells. And then you're going to have your white pulp, which is going to surround your splenic arteries, which are going to form those PALs, or again, it's going to be your periarteriolar lymphatic sheath or lymphocyte sheath. And it's going to contain mostly T cells within that. And then the marginal zone, that's located between your PALs and red pulp. It's going to contain B cells that are organized into your primary follicles that we had mentioned earlier, that eventually develop into secondary follicles upon stimulation. So again, some of the main takeaways here are going to be the red pulp contains your macrophages that are going to get rid of and throw out your old red blood cells. Your white pulp, which is going to be consisting mainly of your T cells, and which also forms your PALs. And then your marginal zone, which is going to contain mostly B cells organized into their respective follicles. And then here's just some histological images that would probably help you within your other courses. And to wrap up the spleen, we're going to talk about a high yield fact that you're going to see over and over again. So for a splenectomy in children, it's going to often lead to an increased incidence of bacterial sepsis. Generally, this is less severe than the effects that it has on adults, although it's going to predispose individuals to blood-borne infections because, again, that goes back to our point that the spleen only deals with blood-borne antigens. And it's going to be especially a problem for encapsulated bacteria. And I would go ahead and ingrain these three bacteria in your brain because you're going to see this over and over and over again. So it's going to be your Streptococcus pneumoniae, Neisseria meningitidis, and Haemophilus influenza. And again, this is going to come up over and over and over again. So I would go ahead and get this memorized. So the next secondary lymphoid organ we're going to talk about is your lymphatic vessels. And one of the really key points that I want to highlight here is going to be how the body drains lymphatics. And kind of a general rule of thumb is going to be having a good understanding of this image. So basically, anything that's over here is going to be drained into your right lymphatic duct, and then everything else within the body is going to be drained within your thoracic duct. And you're probably going to get assessed on a specific body part and saying, okay, if it were in the right arm, where would it drain? Or where if it was in the left leg, where it would drain, and you would say thoracic duct. So I highly, highly recommend kind of getting a general idea of this. They're not going to try to assess you on this obscure areas when they try to assess you on this idea. They're just trying to see if you understand the general concept of, again, if you were draining lymphatics from your right arm, it would go into your right lymphatic duct. And if you were draining maybe, let's say, your right leg, that would go back up into your thoracic duct. And then here is a histological image of kind of how this kind of would sit within the body. And I would highly recommend getting familiar with this image. So the next secondary lymphoid organ that I want to talk about is going to be your mucosal associated lymphoid tissue, or commonly called MALT. So MALT is going to comprise of all your lymphoid cells within the epithelia and lamina propria lying below the body's mucosal surfaces. And there's going to be two main sites that you want to worry about. So the first one is going to be your gut-associated lymphoid tissues, or GALT, which is one of your main sites of your MALT. Your GALT is going to include structures like your tonsils, appendix, and some specialized structures in your small intestine like your Peyer's patches. Your GALT tissue is going to collect antigens from the epithelial surfaces of the GI tract in particular. So again, when you're thinking of GALT, you're thinking about these structures, your tonsils, appendix, Peyer's patches, and then the general idea that your GALT tissues are going to collect antigens from your gastrointestinal tract. The other type of mucosal associated lymphoid tissue, or MALT, is going to be your bronchial associated lymphoid tissues, or BALT. Similarly to the GALT, but they're going to be less organized. And the main concept you want to take away is that your bronchial associated lymphoid tissues are going to be collecting antigens and protecting your respiratory tract. So GALT, you're associating with gastrointestinal, and then BALT is associated with your respiratory tract. And then here's just some extra images to help conceptualize this idea. And the last secondary lymphoid organ we're going to talk about is your tonsils. There's going to be three main types you need to be aware of, and it's going to be your pharyngeal, your palatine, and your lingual tonsils. And then here's just a histological image of what your tonsils would look like, just to help out with that aspect. And that wraps up our secondary lymphoid organs discussion. Again, some of the main secondary lymphoid organs that I would really pay attention to and understand are going to be your lymph nodes, your spleen, how the lymphatic vessels drain, and the different types of MALT. That wraps up this presentation. I would switch over to the PowerPoint slideshow mode when reviewing these practice questions, and it's just kind of going through some of the key concepts or takeaways that we presented in this video.